Okay, so thanks, Micha, for the introduction, and thanks for the invitation to this workshop. And I'm going to talk about sample quality control, importance of sample quality control for proteins, for recombinant proteins, for subsequent measurements, experimental studies. So first of all, I want to give you a short introduction about the reproducibility crisis, the issue that we have, that we all face in biological sciences. And then I will go through you what kind of quality control of recombinant proteins you want to do, why do you want to do them, what can you do, how do you do it, does it actually help, how would you implement such a streamlined process, and how can you further optimize your sample. And we will have a lot of talks about that today, so which means what I will give you is an overview about several techniques that you will hear in much more detail later on. First of all, the reproducibility issue or reproducibility crisis that you, I guess all of you heard before. So the issue is that we are in a core facility, and similar core facilities all over Europe face the same issue, that a lot of time is spent actually on really poor quality samples. And no matter what you do when you put garbage in, you can use a very expensive equipment, very expensive core facilities, you will still get garbage out. So the best experiment will just turn your garbage into expensive garbage, but you don't really get any results from that. So improving the sample quality is one of the essential steps to really get reproducible and reliable experimental information. The international position on that is clear. 50% of the, approximately 50% of the published results are not reproducible. Even what I find shocking is something like 30% of the people state that they can't even reproduce their own measurements. And this is not only a big issue for science, but also obviously for drug design, for pharmaceutical companies, for production of drugs and so on. So this is really a lot of money that goes down the drain. The opinion of researchers with that is very often, I don't have the time to do this quality thing, my boss thinks it's a waste of time, or we did it like that for the last 10 years and it always worked, but sometimes it worked, so let's try. I really don't know how to do that. I mean, this is something that we can discuss, and that's why we're here right now, actually. And a lot of people say, okay, I will do it anyway, let's see, maybe it works. In reality, a lot of times you spend a lot of time, you spend a lot of effort on something that is just not worth doing. So there are people, for instance, who expressed proteins, they had a chromatography, it looked fine, and then only when they crystallized it, they discovered that the crystal had actually another protein. Or similar, another thing, they also got the correct mass for the protein on a page, on an STS page. And only after mass spectrometry measurements, they discovered there were 4 essential amino acids missing that are important for the interaction that they tried to measure, that they tried to characterize. And so on. So there are a lot of things like that where you can see that really you want to know what you're working with. You want to really know in detail what is the quality of your sample, what is your sample really doing. So how to do that and why to do that? As you've heard from Micha, there is ARBRE-MOBIEU. It's a European network about biophysical characterization and resources. And there is another network, which is the P4U, Protein Production and Purification Partnership in Europe. And they both together came up with a minimum sample quality control that you should know, that you should have done. So there is a list of minimum information you should know, and also there is a list of minimum things that you should check with your sample before you go down to more expensive and more elaborate experiments. And what is this kind of information? So first of all, you really want to know your name, primary sequence, concentration of your protein, and also how this concentration was actually measured, and also what is it stored in, what's the buffer composition. And you would be really crazy what kind of samples are brought to facilities. Like people bring samples and they want to have some measurements done with that, and they don't know the concentration, they don't know what the buffer it's in because it's a protein of some colleague that I just bring by and things like that. So you really need this information, you really want to have this information ready. Also, if you have your primary sequence, there is a lot of ways to get more additional information. There are software tools that calculate a lot of things that can be helpful. Most likely you know the ProtParm tool on the XPassy homepage that gives you information about your protein, extinction coefficients, stabilities, PI, theoretical PIs. I also very much like this protein calculator from the Marius Chlor group that also gives you absorption at 205 in addition to 280. I also like the software set fit, which is used from AUC users, which can also give you a lot of information just by pasting your primary sequence of your protein, and you know then things like dN over dC, V bar, and all the above mentioned parameters as well. So what do you want to check at now? So you know about the primary sequence, you know what your protein should be, and you know now you want to figure out what's actually the status of your protein. So first of all, you want to check for purity. Is it really pure? Are there any contaminations? The other thing that you want to check is homogeneity. Is it aggregated? How much of it is monomeric? Is there oligomeric states? Is it a mixture of different species? What do you have in there? And the third minimum check that you have to apply, have to do is the so-called is identity check. So you want to know that what you're dealing with is really the protein that you think you're dealing with. Then there are additional things like, for instance, fold state. You want to make sure that your protein is folded or is not folded, depending on if you're working on intrinsically disordered proteins. And it's always good to know if you actually have a fold state or not. And one thing is, since we're most likely not working with a single batch, you work with batch to batch, you produce the protein over and over, you want to make sure that you have a batch to batch consistency. So you want to check if the next batch actually has the same quality than that batch before. So how do you do that? For purity, what a lot of people do is page, which is obviously one thing, but just keep in mind, if you do a page, whatever you see is proteins. Yeah, so you don't see any other contaminations than protein contaminations in there. And also what you're checking is length of primary sequence. Also, make sure you stain it in a way that you really see impurities. Yeah? If you just stain it with standard Comassi, most likely the sensitivity is not high enough. And if you have like one nice blob here, and the other things are so faint that you don't see them, you say, oh, there is no impurities. Well, actually, there might be. So you really want to use a sensitive way of showing the impurities. Another thing that I recommend everyone to do is, because you anyway do it at one step, is to measure the full UVV spectrum of your protein. At one point, usually you put your sample in a UVV spectrometer and you measure 280 or whatever. Why not just measure the whole spectrum at that point? Because the whole spectrum contains a lot of information. It contains information about scattering. If, for instance, the baseline is not flat, but if the baseline goes up, it means there are already precipitates in there. There is something that is scattering in your sample. Also, you would see, for instance, if you have nucleic acids in there, if the absorbance at 260 is quite high, this tells you something. But there is also a lot of small molecules that would absorb. If you have, for instance, imidazole still remnant in there, or some other small molecules from purification. Yeah? So just look at the spectrum and compare it how a typical protein spectrum should look like. Gives you a lot of information. It's done in 5 minutes. The second thing is, and this is something that Alex Fish is taking much more time later on to go through, is homogeneity. So what you definitely should do after purification, or one step of the purification typically, is size exclusion chromatography. But what does size exclusion chromatography tell you? It tells you different sizes, different shapes, or also if there is column interactions. So you just see, does it come earlier or does it come late? But it's not really that much information, but already you see if there are multiple species, it's not homogeneous, or if it comes in the void, then it's essentially aggregated. Yeah? In addition to that, you can have mols, and what you then really get is you get much more information because now you get mass information. And this mass information is much more accurate than what you would get from STS page or from SEC. And also you see multiple species, and now you can, for each of the species, you can actually say what it is. Is it a monomer? Is it a dimer? And so on, which you would not be able to tell just from the SEC alone. Because here you get mass, while here you get essentially size. The second technique, or that is very easy to do and well recommended, is dynamic light scattering. And also Alex Fish is talking about that later on. And this tells you something about the size. And dynamic light scattering is also very, very sensitive to aggregation. So we also use it to screen for aggregation. And the third important thing is identity. And how do you check if the protein that you have is really the protein that you think it is? And the easiest way to do is intact mass spec. With intact mass spec, you get the full mass of your real protein, and you get it down to accuracy that is much, 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 much better than anything you get on a gel or even SECMOLs or whatsoever. If you're not really sure, and if you have some other things, also what you can do is peptide mass fingerprinting. So essentially, you chop off your protein and look at if the sequence is how it should be. But very often, just the absolute mass of the overall primary sequence is enough. And this is a nice example for a protein, a wild type with 2 mutants, and you can calculate the theoretical mass of these proteins, and with intact mass, just the mass of the overall protein, you see that you measure actually down to a single hydrogen atom, that the mass is actually what it should be. Yeah? So you will easily see if there is a single amino acid residue missing, if you have any post-translational modifications, which you should not have in this case because it's expressed in E. coli and things like that. And there is really, if you're off with the mass, you can already calculate like which one of the amino acids are missing, or if you're way too much off, you have to figure out where is your issue in expressing or chopping down, or maybe you actually have another protein that just looks like if it's similar. Okay, so then in addition, what you can do is measuring fold state. Is your protein folded? One thing you can use for that is CD spectroscopy, where you get information about secondary and tertiary structure, and I'm going to talk about CD spectroscopy actually after lunch. Another thing that you can do, and David Hasselbach is also going to talk about that, is trying to melt your structure. So essentially, you just heat it up and see if there is a structural change upon heating. And if you have a step somewhere in there, then this is a typical sign that, okay, there was a structure to begin with, and it was unfolding. If you don't see any steps, then most likely it was not folded to begin with. There are several methods that you can use to get this information. And last but not least, if you create a new batch, you want to do a batch-to-batch comparison, so you want to make sure that the next batch is similar, and it's easy to use one of these methods, for instance, CD spectroscopy, just to compare the batch to the previous batch, and CD spectroscopy is also recommended because it's an FDA-approved method that tells you really, like, this is the same as it was before, and that's very easy and fast to do. And the nice thing about all these methods that I showed you is, in principle, you can do them all within 1 day. So there is no excuses that I don't have time to do. But does it help? Why would I do it? I mean, it still works, but does it help? And therefore, ARBRE-MOBIEU and P4U set up a questionnaire and wanted to figure out what is the status of the samples that come to core facilities, and how much does knowing this status, is the sample fulfilling the quality control or not, is that giving you any impact on the subsequent experiments? And we had 31 institutions, 47 laboratories, and 186 samples that were imported into this database. Like every time people brought samples, a questionnaire was filled out. And for about 30% of the samples that came to the facility, people said that if you check for purity, homogeneity, and identity, 1/3 of these samples did not pass all 3 tests. So at least they failed either in purity or in homogeneity or in identity. Still, people wanted to run the subsequent experiments. And the subsequent experiments, the downstream applications, from high throughput screening, cell biology, in vivo studies, structural determination, those downstream applications were known for about 130 samples. And people were asked afterwards, did the downstream application actually succeed? And for the samples that failed at least one of these tests, so this 1/3 that we had before, at least one of the tests failed, what came out is approximately 1/5 of the samples succeeded subsequently in their downstream application. So what about the samples that didn't fail, that passed the homogeneity, that passed the purity, and that passed the identity test? For those samples, almost 3/4, so 74% succeeded in downstream application. Yeah? So just checking your sample quality and make sure that your sample is in good shape will bring you from a 25% or 20% chance of succeeding your really expensive downstream application to a 75% succession rate. Okay, so obviously it's worth doing that. So how shall I do that? How can I do that in my own lab? How can I do that with my samples? First of all, you can use literature that are provided. There is SOPs around. And the other thing is you can use facilities. Facilities, as we are in Vienna, ProTech facility, but also here in Brno, it's CETEC. The big facility gives you access to these techniques and gives you actually the possibility to do this quality control. So there are a lot of publications on this case. I mean, you will have access to the presentation, so you can have a look in those publications to see what kind of quality control you want to do, how you want to do it, including SOPs. And actually, CETEC and Vienna BioCenter core facilities, we had an Instruct-ERIC project together. And due to this Instruct-ERIC project, we implemented this protein quality control as a service within the facility. We finished this implementation last autumn, and we already ran some test samples. We ran some test samples in Vienna. We ran some test samples here in Brno. And in Vienna, we're going to establish this service within this year. So right now, we cannot offer it because it still is some things to go. I think also a similar time schedule for CETEC, but maybe within this year, the service will be offered. And what does that mean? The service would mean that you bring your sample, and what we do in Vienna, for instance, we do UV-Vis, STS page, DLS filtered, unfiltered. We measure secomols, intact mass spec, and thermofluor, and we do that on 5 to 10 samples at the same time, all streamlined, and we do that in one day. And on another day, we have everything analyzed, so within 2 days, we can go between 5 to 10 samples, and you will get a sheet that tells you, okay, which of the things, purity, homogeneity, identity, and so on, did you succeed, did you fail? There will be a table of calculated values that you get of measured values, concentrations, homogeneity properties, and so on. And you will have subsequent and also the results of all the techniques in more details. So in principle, if your sample is in good shape, you check it, you have the sample quality controlled, everything is great, you go on with your expensive X-ray or cryo-EM or whatever, but what would you do now if your sample is in bad shape? Because that's obviously what you will see quite often. What you then need to do is you need to do optimization. And optimization is another thing that we're going to discuss during this workshop today. I just want to briefly give you an idea of what you can optimize or what you want to try. So first of all, you want to try different buffers. So David can, I guess he will show that later on in his lecture, but the buffering substances themselves actually do make a big difference. Also, you can change pH, you can change different salts, ions, you can use detergents, cofactor ligands to your protein, you can choose different concentrations. You really want to screen a lot of variabilities, a lot of variables to see which of those will affect your protein. What kind of screens do you want to do? What kind of optimization? Sometimes you don't even get your protein to begin with. You actually have to express it in insoluble inclusion bodies, and you have to refold them first. So you want to do refolding screens. Another thing that you want to do is you want to have stability screens. Your protein is here, it's fine, but for how long? You want to see, does it unfold over time? Does it degrade? Does it aggregate? At least you want to have an idea about how stable is your protein due to the course of your experiment. So if you're running an AUC experiment that takes 24 hours, or if you're running NMR experiments that take in several hours, you want to make sure that within that time your protein actually is not aggregating. You want to see temperature dependencies, at what temperatures is it fine or not. If you freeze and thaw it, if you store it, is it actually happy after that as well or not? Things like that. So those are things that you really want to screen and check. Another thing is crystallize ability. I'm not talking about this because there are many more different screens and it's a lot of automation in there. But this is not such an intelligent design, I would say, but really just a gunshot approach. Another thing is you want to screen for homogeneity, under which condition your sample is actually very homogeneous. And unfortunately, up until now, there is not really a high throughput method to look for homogeneity. Maybe David Hasselbach showing us then the mass photometry. I think this might be a method that you really might be a high throughput method for homogeneity screening soon. So what to use for screenings? There are 3 techniques that we have and that are easy to use. You can think of others as well, for sure. But one of them is thermofluor, which essentially just uses a fluorescent dye and a real-time PCR machine to tell you if your protein unfolds or not. Then there is nano-DSF, which measures tryptophan fluorescence and can also see if your protein is unfolding or not. And then there is dynamic light scattering, which tells you the size, essentially if there is how big is your protein and if it's diffusing. And the advantage of that one is you don't need any label whatsoever. Here you need at least tryptophans, and here you actually have to add dye as well. So I'm not going to talk about the thermofluor in more detail because David is covering that. But you will use the nano-DSF within this practical course. So I will shortly describe this, and also you will use the DLS in this course, and I also shortly show you how this can be used for screening conditions. So the nano-DSF is, there is one instrument that you will see, that's the so-called Prometheus from Nanotempor Technologies. And essentially what this instrument is doing, it measures tryptophan fluorescence of the protein at 330 nanometers and at 350 nanometers. And the idea is that upon unfolding of your protein, the tryptophan fluorescence is going to change. Because in the inside of the protein, and where it's hydrophobic, and the exterior, where it's hydrophilic surroundings, the fluorescence will change. And that's how you follow that, and you might see an increase or decrease of fluorescence. So those are real measurements, and as you see, the tryptophan fluorescence goes down with temperature overall, but then you have these steps. And this is a nice thing because you see 2 steps, and that was a fusion protein with 2 domains. The instrument looks like that. It works with very low volumes. You only need about 10 microliters. You can run 48 samples at the same time. You can also either do thermal unfolding, or you can also do unfolding with different caltropes, like guanidine chloride, urea, or whatsoever, just to see under which conditions your protein is most stable. Okay. And what we use, we don't have the Prometheus, unfortunately, in Vienna yet. Hopefully we'll get it this year. But we use is the DLS plate reader, and we use that to really figure out which of the buffers prevent aggregation, or in which of the buffers the protein is most happy. And the advantage is it's label-free, so you don't have to add anything. You need about 6 microliters per sample if you go into a 1536 well plate, so that's really very low volume, and concentrations are also reasonable. And what we typically do is we measure time-dependent aggregation. So we have, for instance, 96 different conditions. We have buffer mixtures, whatever you prefer, pH screens, salt screens, and so on. And then you measure, for instance, the size of your sample, and you see that for each of the conditions, of the 96 conditions, you get different sizes. And that already tells you, okay, in this buffer, it looks bigger, which means there is more aggregates present than in another buffer. And what you clearly could see here is that in this row, the size was really, really much smaller than on all the others. And what we concluded from that buffer screen is this was the only screen where detergent was present. So the protein obviously needed detergent to be happy, and then in a subsequent screen, we screened different detergent molecules. But also you can not only measure it as is, but you can measure it over time, over 2 days, for instance, over weeks. We also measured, for instance, antibody stability for a month, where in the meantime you store the plate and bring it back to the plate reader and measure it again, and you can really see deterioration over a long time. Okay, so to sum up, I want you to grasp the idea of protein quality control and what are the 3 minimum criteria that you want to check. You want to check for purity of your proteins, homogeneity, and identity. In addition, what sometimes might be interesting is the fold state, and definitely you want to do a batch-to-batch consistently. So I definitely would say you do as much as possible on it, but at least those 3 are the minimum criteria that we recommend in ARBRE and P4U. And once you figure out that your protein is actually not happy and it's not homogeneous and it's not in a good shape, you want to do optimization. So you want to buffer optimize for whatever you want, refolding, stability, crystallize ability, and so on. Okay, so that's my group, where Peggy is still here, which unfortunately she left in December. And I want to thank you for your attention. Thanks.